Welcome everybody who are going to use BioIllustrator program. In this video, I would like to demonstrate the functions of this program. This program is aimed at scientists, students, amateurs interested in the creation of illustration for scientific articles, PhD or diploma theses, different presentation on different topics of biology. The drag and drop function of curated templates in this program saves your time on the creation of professional scientific illustration. Of course, this program provides standard tools for the manipulation of images, texts, geometrical shapes, and lines. The current version contains templates for the following fields of biology: molecular biology, cell biology, microbiology, and botany. Each field is divided to subfields that facilitate the search for the specific template. The biology field and biology subfield you can see on the left side, and below you see the templates related to the corresponding subfield of biology. Besides the subfields of biology, there are also templates of laboratory equipment, which can be quite useful for creation of illustration of your workflow. At the right side, at the upper corner, you can see another type of templates. It's so-called bio-shapes. If you, let's say, change the corresponding field, you can choose the different subfields, and automatically you also change the templates, bio-shape templates, at the right upper corner, which you can see, for example, here. Now I would like to make illustration of part of the cell, and by this way we will see how we can use all templates to make this particular illustration. First of all, we need templates of cell envelope. In this case, we just go to the cell components and we can take phospholipid B layer. We simply click and drag and drop on the canvas. This image is small and we would like to make this image on the whole canvas. So to do this, first we should double click, and by doing this we select this image and you see the green box around. Now if we click and drag, we can increase the size of this image. Without selection, without this green box, if we again click and drag, we can move the image on the canvas. Let's make it a little bit bigger. Double click and click and drag. Now we have a little bit bigger, enough for this canvas, and shift a little bit down. So next, probably we need some nucleus. We can use two types. Let's take this one. Again, click and drag and drop. I think we should also increase a little bit size. Double click and drag, and we will increase. And without selection, click and drag, we can move this. So we can move somewhere like this. Let's say we also need some receptor. Then we should switch to molecular biology field, and then we can switch to the receptor and transporter. Here, among these subfield images, we can take, for example, notch receptor, this heterodimer. This template is a little bit big. We will decrease the size and shift. What we can do now, we can turn this receptor at the right angle. To do this, for people who are using touchpad, we should click two finger, and then you see the sign of rotation, and then we can rotate by shifting one finger. There is another possibility to turn the template. For this, we need to select template, and then we can use this button to rotate the selected image. It's also applied to text in bio-shape template. As you see, we can turn this. And here, if you type the number 0, then it also will be applied to this selected template. So come back. Okay, this is the template as image. On the right side in the upper corner, you see the corresponding templates as BioShape. For example, we can use generic receptor, this one. In this case, you have more options for different colors for lines and also different colors for filling this image. To do this, again, double click, and then here below this upper corner, you see the different options for modification of this bio template. We can open color table, and then let's say we choose the green. Maybe we need less opacity. Then if we select now stroke color, then we can change also the color of lines. We select again green. Then we would like to put this through the membrane. If we need, we can increase the size in the same way as we did this for templates taken from this. Then we can also use ligand. We can fill this shape. We should press this button, then use the specific color, let's say red one, and we can put somewhere here as ligand for this generic receptor. So this is the advantage of this BioShape template, that you have flexibility for different colors in comparison with images on the left side. With these images you can increase size, you can turn at right angle, but you cannot change content. You can change also the opacity of the image. For this you have to select image and then use this slider just to decrease or increase opacity of the image. Besides the BioShape templates, the program has also basic shapes. If we press this button, we see the list of these shapes and also lines. Let's use these lines. We know that notch receptor can be cleaved, and cytosol part located here then move into the nucleus. At the left side we have this cleaved part of notch. We can again make it a little bit smaller, and then for example we can use this line, drag and drop here. If we double click on this line, we see 3 green dots. If mouse pointer on top one of this green dot, when we click, we can drag, and then you see we can change the curve. And by using this line, you can double click, we can show the pathway. What we can do, we can again select this line and then use this option for modification of this line. We can add at the left side, we can add arrow, and we can increase the width of this line. We also can use different types of this line. This of course is quite useful for different pathways. Let's say we can use another one, modify this as we wish, just to show that this part of notch is going to be inside of the nucleus. Again, we can use this style, and we will put the arrow at the right end of this line. And of course we can change the color of this line, again using these buttons. To insert the text on the canvas, we should use this button. When we press, we have this text. We can move this text in the same way as we did this with images, just click and drag. Let's say we will write that this is notch receptor. We can change the size of the letters. To do this, we should press this button when we select the text. we can change the font size. 40 is too small, 18. In the same way as we did with image, we can turn this label if we would like. Also, we can, for example, use some basic shapes like node. We can add some label. We can write that this is generic receptor. So we press text button and then we can change this. When illustration is completed, it can be saved to graphical files or to custom files which can be used later for editing. We open save function. Let's close this. As you can see here, BioIllustrator document. This is the custom file. So let's save this illustration to the BioIllustrator document. Test. Save. Then let's close this file and then open again. So we have our test file, double click on this file, and then we have open the illustration which we prepared before. As I said, this illustration can be saved to the graphical file. For this, we just use export function, and then we can switch to any, let's say, TIFF image, test2, and save. Here you see the test2 file, and because this is a graphical file, user can open this with different program, and we also can open this with BioIllustrator. If we do this, then we have second document, test2, and here in the middle you see this illustration as image. Now let's switch to the test document again. There is another option. Besides of saving your illustration to the file, what you can do, you just have to click on the canvas, two finger on the touchpad, copy whole canvas with your illustration to the buffer. And then let's say we open Keynote, then we can use pass function to pass this whole illustration. Let's say we don't need whole canvas, we would like to take the only part of this illustration. Then you click on the canvas, make this green box around, click again on the canvas, two finger, and copy selected part of the canvas. And then again you can paste this part of the canvas. So now you see here only the part of this illustration. The same way you can use to insert some images or shapes from Keynote, let's say animals. If you select, copy, then paste this to the canvas. We'll put here our cow. Besides of interchange between different apps, of course, you can simply drag and drop any figure from collection of your images, some cells, simply drag and drop here. You can do by this way, or you can copy this file and paste again. At this point, I would like to finish this first part of this video, and I will continue in the second part of the video.